So we're so pleased to welcome everyone to our genetic research education series with a focus on SOD1. We are Genetic ALS and FTD and the Legacy. You might be wondering what that is. Well, we are a patient-led organization proud to serve our community. We say "our" because us and our leadership team are people impacted by genetic ALS and FTD. For instance, my family has a C9orf72 repeat expansion mutation. We've lost many relatives to it, and I carry this case. So we strive to serve the needs of the community through education and support resources like today's webinar. And we also serve to represent our interests of this community in dialogue with the academic field studying us, our government, and the industry developing drugs for us. We just want to start this webinar off with some acknowledgments. First of all, to our speakers today, thank you all so much for giving of your time to share your stories. We want to thank the investigators, companies, and universities undertaking the research that we will discuss. We want to thank all of the people in our families that participated in research preceding us that allow the ever finer knowledge to power the next breakthroughs. So, in my family, we've had 2 generations of brain donation. And I know in many other families that's existed, and that's wonderful. We want to acknowledge that. And we want to acknowledge our gratitude to our sponsor for this series, Biogen. And with that, I will kick it over to Tara Jones, genetic counselor. Tara, take it away.
All right. Thank you, everyone, for having me. I'm excited to be the genetic counselor who's presenting on genetics for this series. I do apologize if some of these topics are a little bit of an overkill. Some of you may not be as familiar with genetics and genetic counseling. I do want to let you know I do have some conflicts of interest. And I'm going to skip through the agenda because I don't have much time here. So why do you need to see a genetic counselor? So if you are affected with ALS or FTD or another neurologic disease, your neurologist might be willing to test you, especially if they don't have a genetic counselor on their team. But for those who are asymptomatic, a lot of times providers do kind of require that you meet with a genetic counselor first. So just so you're kind of aware of what we do, each new patient session lasts about 45 minutes to an hour long. And we will educate about basic genetic concepts as well as specific neurogenetic concepts. We will be taking a very complex family history and providing a risk assessment based off of your personal and family history. And we'll at the same time kind of be assessing your risk perception and your motivation for testing. We'll have an in-depth discussion on whether genetic testing seems to be right for you in your current life stage. If it's looking like, we might move forward, we'll talk about test procedure, as well as alternatives to testing. And we help you come to a shared decision if that's what would be helpful. Some people do enter a genetic counseling session already knowing what they want, and we have very little influence on that decision. And we're really there to kind of facilitate testing and make sure that things are going smoothly. And then the results are in a separate session. We will do an interpretation of those results. We'll meet with you to disclose, and we'll provide some next steps so that we don't leave you hanging. And it is important to know that you don't need to want testing in order to meet with a genetic counselor. Now, in that discussion, there's a lot of things to consider. I don't have time to go into these in detail, But I do want to highlight the research aspect. So for observational research, you might not actually need to know of your own genetic status. However, for interventional research, where they might be trialing a drug, you will likely need to know your genetic status for that. All right, going into genetics basics. Believe it or not, this is one of the simplest images I found of the central dogma. You might remember DNA to RNA to protein, and I do kind of correlate that to baking. One thing that you might not be as aware of is RNA to mRNA, where these green introns are spliced out. You can kind of think of that as your own personal touch to the recipe. Let's say you're out of eggs and you substitute applesauce. Now, what happens when there is something wrong with that recipe? So the genetic code is kind of read in these 3-letter words, so to speak. And if we were trying to spell out the car was red, if we swap that E for an O, the car was rod. It just doesn't make sense anymore. But let's say, for example, instead of an O, it was swapped for an A, and it says the car was rad. That still is a comprehensive sentence. And so that's kind of an example where changes in our genetic code might not always be pathogenic. They might actually be benign, and it might just be a different way to talk about the car, for example. And here's some other examples of other types of mutations or pathogenic variants. As well, I'd like to talk a little bit about inheritance. So specifically for SOD1, there are 2 different types of inheritance. You can we have 2 copies of the SOD1 gene. One copy we got from the sperm and one copy we got from the egg that were fertilized to conceive us. And so with a dominant condition, you only need one copy to be broken in order to put someone at risk of disease. And so common themes here, males and females can equally be affected. And because you have one broken copy, one working copy, you basically have a 50% chance to pass that down to each pregnancy. And so each child of a parent who does have a SOD1 pathogenic variant has a 50% chance to also inherit that variant. Now, there's also autosomal recessive inheritance with this gene, though it's not as common. There is a case, a specific variant in SOD1 where having 2 broken copies essentially will guarantee if you live long enough, you will develop disease. But having just one broken copy and a working copy just increases your risk to develop disease, and it's not a guarantee. And so in these cases, again, we'll still see males and females equally affected. It might appear that there's only individuals in one generation affected. And this gives the appearance of skipping a generation, but in reality, the genes aren't skipping. It might just be kind of the penetrance that appears to be skipping. With recessive conditions, they might be earlier onset. They might be more common in certain ethnic groups. And sometimes parents might be related by blood, which increases the chances that they're both considered to be carriers for the same condition. So in this example, the mother has 2 broken copies and therefore can only pass down a broken copy to each of her children. So those are kind of the genetics basics. And now kind of going into how to read your genetic report. If you've ever had genetic testing, hopefully your provider has shared a copy of that report. If they haven't, definitely go back to them and ask. If you remember the name of the lab, go directly to the lab and ask them for a copy of it. There will be a lot of stuff on a genetic report, and they all look different depending on which lab did the testing. But the most important pieces are going to be the kind of classification of the genetic variant, which gene it was in, which is usually italicized in all caps, the specific change that happens at the DNA level, and then what happens to the corresponding protein or that final cake if we go back to our recipe analogy. And so a pathogenic variant, that's kind of the classification where this is pathogenic, it's clearly contributing to disease. In a gene MFN2 or mitofusin 2, this is not a ALS gene. And this C dot nomenclature is basically what's happening at the DNA level. So at position 281, where most people have a G, instead this person has an A. And what happens to the protein in that case is at position 94, instead of an arginine, they have a glutamate. And that's kind of along the protein chain. You might also see additionally a uncertain variant, maybe in a different gene. And we'll kind of go into a little bit more detail on what uncertain means. So essentially, just as it sounds, an uncertain variant, we're not sure, does this change cause the gene to not work properly, or is it just a unique difference in you? Again, thinking about the car was rod versus the car was red or rad. We still might not have any major consequence with that genetic change. And so in these cases, it does take a provider who's knowledgeable in genetics to do a lot more digging and kind of decide, how likely is it that this change is actually causing the gene to not work? And in this case, it might be helpful to test other family members to gather more clues, but not for any sort of predictive purpose. And then finally, just kind of a brief slide on SOD1 specifics. So if you do have ALS and you have a family history of disease, then there's a 15 to 20% chance that you test positive for SOD1. Whereas if you have zero family history of disease, there's about a 3% chance to test positive. So if we look at everyone with ALS across the board who gets tested, about 3% will test positive in SOD1. And that's about all I have time for today. So I wanted to thank you for your time and provide my email in case anyone wants to reach out to me.
Oh, there I am. All right. Hi, everybody. I'm Cassandra Haddad, and I got too excited to hit the camera button too many times. So hopefully you can all see me and see my slides now. So thank you all for attending tonight's webinar. I'm thrilled to see you all here tonight, and I look forward to sharing some real-world experience with you. I'm Cassandra Haddad, and I come from an SOD1 A5V ALS family, and I'm a genetic carrier. For at least 7 generations, my family has been decimated by SOD1, which is a particularly rapid genetic variant. Here we go. So the first picture is my grandfather, Harold Lindsay, and he passed away in 1990, just before our gene was identified. For many years, we thought the rest of our family had escaped this disease that's claimed the lives of over 33 family members, until my uncle, the second picture, Dwayne Lindsay, noticed some leg weakness. He was able to get genetic testing, and that's when we learned the cause of mutation in our family. He was a participant in the Valor trial. However, his trial participation was impacted by the pandemic, and he did pass away in 2020. The next picture is my mom, Cindy Weber. The day my uncle died, my mom's symptoms started. She participated in the Healy trial as Valor had closed and was the first person in the country to receive tofersen through expanded access. She lived for 25 months, which is really incredible for our family. Last week, I actually received a phone call asking if I wanted to know her status in the Healy trial. She was on a drug that was canceled for futility, but she had been getting active drug, and we do believe that drug was helpful for her. So it was just a cool follow-up to kind of find out which arm she had been in. The next picture is me a few years ago at my very first advocacy events with my twins. And they could be the 9th recorded generation at risk. As a genetic carrier, I know research and advocacy is the key to making ALS a livable disease. I want to help the ALS community who is currently impacted, those at risk like myself of ALS or FTD. And despite working full-time as a nurse practitioner and having 7-year-old twins, I see research and advocacy as something that I must do rather than I choose to do. Not only for everyone with ALS today and to try and save myself, but my biggest motivation is to try and save my at-risk children. To this end, I'm the vice chair of End the Legacy, and research and advocacy are 2 of our core pillars. As part of the impacted community, our voices must be heard in the development and implementation of trials for us and for our family members. So now onto my participation in research. I'm involved in 2 different types of trials, observational and interventional. Observational means they basically watch you. They just observe you and try and unravel some of the mysteries of ALS. Interventional means at some point they do something to you rather than just watching you. I'm currently enrolled in 2 observational family trials and will be joining a 3rd next month. And I'm also in an interventional trial. So first I'll talk a little bit about observational trials. They're not all bad or completely painful, although there are definitely some painful parts. So this was my first observational trial experience. My mom had been recently diagnosed, and so understandably it was a hard time for my family. But my sister and I went to the city where this trial is together, and we made a whole weekend of it, and we had a really good time. And this was really important to my sister because she's gene negative, but it made her feel like she was empowered to advocate for me and to be involved. Because this was during the pandemic, this was a real weird time to get into a trial. So I had a couple Zoom appointments first and actually found out my genetic test results on Zoom. And then when I went to the in-person visit, I had blood taken, a skin biopsy, an EMG, and a spinal tap. These range from mild to medium in unpleasantness. But a few hours after all those tests were over, we were out on the town and out in dinner. The one downside of participating in a lot of observational research trials is they are not funded. And so you pay for all of your own travel. So that's definitely a barrier. The next observational trial I joined happened to be like the following month. So I had a lot of back-to-back travel, but that was kind of my choice. This was through a different organization, and it's the one that I'm still currently the most active with. And actually the first and second trial I joined are actually working together now to decrease kind of the burden on participants and share information, which is great. So I go to this town twice a year. These first 2 visits for this trial happened to overlap with the interventional trial. So I was very fortunate in that the interventional trial covered the cost of these trips. But that doesn't always happen. So yes, the appointments are long and they involve poking and prodding and a lot of the same tests I had just had done. But someone always goes with me when I travel. And in this case, it's my best friend of 25 years goes with me to this town. And we have a really good time. It's actually turned to one of my favorite cities in the country. It's hard in that I leave my children for, 2 or 3 days and I take some time off work. But my family and friends have been really supportive of me trying to use this as some downtime and some positive time to kind of regroup and just have some time alone from and away from the hustle and bustle of life. I also use this as a way to spread awareness when I travel. I usually wear like logo t-shirts and talk to people in the airport and try to advocate while I'm there. So that brings me to the interventional trial. So I did mention it covered my first 2 visits for my second observational trial. And it's the only pre-symptomatic genetic trial currently going on. And basically what that means is it involves a lot of waiting around and hoping that nothing happens. It's absolutely the best thing I feel that I have done for myself. And honestly, it's kind of the only thing currently that I can do to even try and intervene early and figure out exactly if I get ALS when that happens. So the trial consists of those 2 visits I mentioned to the center, but after that, all the monitoring is done remotely. So that's pretty amazing. The trial fits easily into my life because every month a traveling phlebotomist comes to either my work or my home, whichever is most convenient for me. They draw 2 tubes of blood, they ask a couple questions, and that's it. I have a monthly Zoom call with a nurse practitioner, and it literally takes less than 5 minutes if we don't get chit-chatty. So I often do that like in between patients or over lunch or sometimes from the car. And unless you develop symptoms or that blood test, which is looking at neurofilaments changes, you don't have to travel again for the duration of the trial, which is like 4.5 years. So I'm already halfway through, which is flying by. None of the trials, including this one, tell you your actual results. This trial only says, yes, you're below threshold, or no, you're above. So that is one thing I wish was different. And then if your blood level goes up, what happens is you get randomized into a placebo-controlled group, meaning some people get drug and some people don't, and they try and see if they can use this drug to prevent symptom onset. However, if you do get symptoms during that period, or if you don't get a rise in the blood level and you just get symptoms, then you automatically go on to drug. Although for our gene, that is pretty rare. And you know, the science is still unfolding and changing, and we don't have all the answers yet, and that's part of what participating in this trial is all about. So all trials are different, and what works for me may or may not work for you, but they can fit into your life, and I do think it's absolutely imperative for me to participate in research. There's a lot of really exciting developments coming this year with some new funding that's going to decrease travel burden and financial burdens, and be a little bit more creative with like some decentralized trials and get more people involved into research. This final picture is from the International Symposium that I was so fortunate to be able to attend on behalf of the group in Basel. And right here is myself and 2 other SOD1 people who are actually impacted by ALS. So Paula's from Canada and Helga is from Iceland. We all have different mutations, but we really bonded and we called ourselves the cousins, even though we're actually not related. And one of these ladies actually had almost 100 treatments with the only approved drug to treat SOD1 ALS, and she's doing really well. And meeting her was absolutely incredible because it gave me so much hope that due to her participation in research, that we might be able to survive ALS. And that's the goal for all of us, is to make ALS a survivable disease. So I hope, you know, a little glimpse into my life participating in research was helpful, and thank you so much.
Hi, I'm Julie Granning, and let me get share screen here. Also share audio, entire screen. Yes, that one. Second. There, did I do that right this time? You can see my screen and hear me? Amazing. Hi, I'm Julie Granning, and let me get you over. Here we go. So, and someone stop me over text or something else if this isn't working. I lost my mom to ALS in 2013, and the 27th of this month will be 10 years, obviously, or 2014. So it's 10 years this month, coming up in just 4 days, which is a little difficult. I am one of those people who we had no family history that we knew of. My mom had two main doctors, and the younger one kept saying, I see things that are telling me that this might be genetic. I think it's just worth doing the testing. And the older doctor was like, no, this is sporadic. There's no family history. So we didn't do genetic testing for my mom. She, about 18 months, took her very, very quickly. We were able to, come home with her, and it was a very hard time, but we weren't really thinking beyond my mom at this point. And she even missed my wedding. She was actually still alive, but she had her own party because she could not leave the hospice care at that time. But she passed in 2014, and I moved on with my husband, and we made a life, and everything had been going really well until my dad, for fun, bought a DNA kit. And my mom, my grandfather was 100% Italian. So we were joking, well, I get, around 25% Italian, and I had 0% Italian and about 33% Polish, which shouldn't be in there. And I started asking questions, and I started contacting these family members, these DNA matches. And one of them said, I'd love to work with you. I'm trying to piece together my mother's medical family tree. And this is what I got back, because the entire family was full of ALS. So obviously this was very difficult. And this is the point when I called a place that had helped when I was a caregiver to my mom. And the green arrow is my, I guess, biological grandfather. He passed without developing ALS, but in his 50s. And my mom is not actually on this chart, but you can see just how prevalent. They pointed me toward a genetic counselor who I was able to talk to and talk about decisions and finding out and studies and what would be the pros and cons of all of that. And to me, the biggest pros were my kids, because I didn't get choices in family planning. I already had 2 kids, and it was heartbreaking to know that I have a 50% shot of passing this on to them. So I now participate in a couple studies. I was doing one online where I think it was recording speech. I have to go back to that one. The main one that I'm doing right now is one of the big observational studies. But as Cassandra noted, it's very difficult because traveling can be so cost prohibitive. And I'm really looking forward to getting to a point as a community where there is funding for this kind of thing, because right now the only people able to participate in these studies are people who live locally and people who are fortunate enough to be able to travel. And so far I've only been able to make it to one, and I'm hoping to do a second one. And at the second one, I have signed up to begin doing spinal taps so that they can measure levels, hopefully, of NFLs. Although I know that in that one, I will not actually be receiving results back. To me, it's important to my family, because anything I can do that would, give any information for interventional studies in the future, for ways we can fight this in the future, to me, that comes down to helping these guys. I love my kids, and to me, it's going to be all about them. And I'm hoping to maybe get into an interventional study coming up. They've got some new ones coming out, and some people are exploring some new ideas of, past drugs that have worked on ALS patients, and what would they do in the pre-symptomatic? Would they prevent? Would they slow down onset? There's a lot of things that are hopefully going to be coming out in the next 1, 5, 10 years, so that hopefully my kids aren't having quite the same problem that I have been. Let's see, there we go. And share. Thank you. And I hope that was helpful or informative in some way.
Hello, everybody. This is a very tough act to follow. I should have gone first. Amazing work, you guys. We put it in presenter mode. Can you see my slides? Hope you do. So my name is Senda Ajroud-Driss. I have a very strong interest in patients who have familial ALS. I've always been interested in neurogenetics. And I had the fortune of really following many patients with SOD1 genetic ALS and getting to know them on a very personal level. So I think I'm not going to dwell on this because it was already presented. Tara did an amazing job talking about those things. But it's interesting, and this too, we chose the same kind of nice drawing to describe how the information that we have in our DNA in the nucleus, which is really the headquarters of the cell, gets all the way down to the cytoplasm and help make the machinery that make the cell function, and then by that make our whole body function. And you could understand very well if there is a faulty information or a mutation in the DNA, how that goes all the way down to the protein. And when that messenger RNA, when the DNA is transcribed from the DNA to the RNA and then packaged in this messenger RNA and shipped to the cytoplasm, and then it gets translated into a protein, what it starts actually is, it's like thinking of beads. The amino acids are beads on a long chain. And those beads, this chain will change in conformation and make the actual protein. So what does the SOD protein looks like? This is the actual SOD protein. And it's like this because there is specific information in the beads of the chain that make it have this shape. What happens when there is a mutation in this protein, when there is a mutation, and there is about 200, over 200 mutation that we know about that can cause disease, the conformation of this protein changes. Imagine that you have a string, and in that string there is a kink, and you're trying to wrap it, and it just doesn't wrap adequately because of that kink. The mutation is a kink, and it prevents from the protein from folding, and we call it a misfolded protein. And that misfolded protein, what it does is actually it doesn't stop making what it's supposed to. It's an enzyme, but it acquires a new toxic function. And we call that a gain of a toxic function. We knew very early on that from the mouse model, that the more abnormal SOD1 you give the mouse, the more early the disease happened and the faster the progression. It was very clear that this protein, although we don't know exactly how specifically it causes ALS, it is obvious that this misfolded protein with this gain of toxic function, the more of it, the best way to kind of help with it is to try and reduce that level as much as we can. Of course, we don't want to turn it off completely because we need some function of that protein. But if we try and decrease it level, we will help with the disease. We'll probably help with the progression of the disease. So this is basically what, and this is from the Biogen website that is trying to explain how an antisense oligonucleotide works. So as I explained earlier, you have a normal DNA, you get a normal protein, you have a mutation, you end up with an abnormal RNA, and then you end up with a toxic protein. So what if we decrease the production, stop the production of that protein? And one way to do it is with antisense oligonucleotide, or ASO. It's actually a piece of synthetic DNA. It does not interact with your own DNA at all, but it will interact with the RNA and will stop that RNA from going to the protein level it will degrade it basically. And what that will cause a drop in that protein level. So I really cannot stop from showing this, and I'm sure many of you have seen it already, but this is the paper that described the trial, the VALOR trial, that led to the approval of tofersen for the patient who have SOD1 ALS. And I really want to show you this. I'm going to tell you about it very quickly. So the study is, they used the second generation ASO. The first one was tried in 2013. And the trial, the placebo-controlled portion of the trial lasted 28 weeks, a little bit more than 6 months, almost 7 months. The first patient was dosed in 2019. They dosed 108 patients, and they had to have disease. This is a study for patients who had ALS due to SOD1 mutation. And what they did, 60 of these 108 had a specific mutation that caused the fast progression of the disease, like the A5V, for example. Every single mutation in SOD1 causes the progression to be a little bit different, from the A5V that causes a fast progression to the D90A that causes a super, super slow progression. And so they did that. And the reason to choose patients who had rapid progression of the disease, thinking if there is a big effect of the drug, we will see it very quickly. And the primary endpoint was the change in the from baseline in the ALSFRS at week 28. The secondary endpoint, they used the level of SOD1 because they needed to know if they're trying to stop the protection of the SOD1, they want to see that it drops down. And they looked at the neurofilament level in the blood. And I'm sure you've seen this already, but I have to go over it again. And this study did show at the end of the 28 weeks that actually the drug did what it's supposed to do. It dropped the SOD level. It started by 12 weeks. It dropped it by about 30%, and it dropped the neurofilament level. So we know now that this drug did what it's supposed to, turned off the production of some of that SOD1, and using neurofilament as a surrogate for the health of the motor neuron, it tells us that it decreased the neurodegeneration. And we can see the second part of the study. So the blue are the ones who were on placebo, the red are the ones who received active drug. And we can see that at the end of the 28 week, when the patient were switched to the open label, the ones that were on placebo, the same thing happened. The SOD dropped, the neurofilament dropped. So, however, the primary endpoint of the study was the change at 28 weeks of the function of the patient. So at 28 weeks, although there was a little bit of difference between how the patient who received the drug and the patient who didn't, who were on placebo, it wasn't significant. It's only when the study went all the way to 52 weeks that we can start seeing the difference, and it's appearing more in multiple assessment, like the breathing and the motor strength. During the last the International Motor Neuron Disease Symposium in Basel, Dr. Sukovich showed the result over 2 years. And not only she showed that 25% of the patient actually showed an improvement, which is just an incredible finding. Not only are we slowing the disease, but we are watching some repair, which is great. Based on this, the study received conditional approval because the effect was obvious on the neurofilament. The effect at 28 weeks was not evident on the function, unfortunately. Of course, this study had side effects. The drug was given intrathecally, that means with a spinal tap to go into the spinal fluid, and every single patient had some sort of side effect, usually a post-LP headache. But in 7% of the patient, there was a risk of chemical meningitis and increased intracranial pressure. That's why now when we do the study, we always follow the result of the spinal fluid to make sure that the patient do not develop a chemical meningitis. We do see that the protein and the cells in the spinal fluid do go up, and that was noted in the study, but we'll really watch for a headache that doesn't go away, nausea, vomiting, trouble with vision that may suggest increase intracranial pressure. So that is very exciting, but it's not the only thing that is happening. I think the SOD1 problem is an easy problem to fix. How are we going to turn off this gene? It's not very difficult. We can do it through a DNA, like ASO for antisense oligonucleotide, but we can also use a small RNA that will interfere with the actual RNA and block it. We do that by the addition of a virus. If we put that RNA in the virus and inject it, maybe all we need is one injection, and we don't need the repeated injection. For the person, we do have to administer the treatment every 4 weeks. Maybe we can just have an intrathecal injection of the RNA by itself, the small interfering RNA. Or I'm sure you've all heard about CRISPR, and CRISPR is a way for the bacteria to actually protect themselves from the invasion of certain viruses or certain microbes. And they have this tool to edit the genome, and that could be harnessed. And can you imagine? We can go in and change the mutation or cut it, replace it, and do many things about it. We can also, using viruses, we can deliver a new gene. We can add a gene that will enhance, for example, the neuronal protection or make growth factor that will help with protect the motor neuron. So I'm going to tell you a little bit about these study and where they are. Well, CRISPR, it's only in the animal, but I wanted to show you this. This is a study done at Northwestern by Dr. Dang, and he used CRISPR to edit the genome of the mice that are carrying an SOD1 mutation. And when you start pre-symptomatically, you can prevent the disease from happening. These are the mice, the control mice, and these are the mice who actually received the CRISPR treatment, and they don't have disease at all. So this is really amazing. The problem with CRISPR is we need to figure out a way to deliver it to the motor neuron. So delivery is going to be an issue, and a lot of scientists are working on it. So there is a study, let's see, I don't see my, yes, there is a study that is, as I told you, looking at injecting a microRNA that will turn off the SOD or decrease the level of SOD1 using a virus. The study is sponsored by Unicure. It's supposed to be a phase 1 and 2 multi-center 3-part study, where the first part is a dose escalation study to know what the actual dose is. And then there will be a placebo-controlled phase 2 study and then an extension. They haven't started yet. And the injection of the virus will be an intrathecal administration. When I say virus, I hope I don't scare you. These are engineered viruses that do not cause a disease in human. The genetic material of the virus is removed, and the genetic material, like the RNA that we want to be expressed, is placed there. And as I said, the study hasn't started yet, and I don't know where it's going to be, but it's going to be a US site will be involved in this study. This is another study sponsored by Arrowhead Pharmaceutical that involved the intrathecal injection of an interfering RNA, an RNA that will actually block the RNA of the SOD1 gene. And also, as I said, this one is going to be a phase 1 study, and it's intrathecally administered. So we don't know yet when this study will start soon, but we don't know about the sites yet. This study, on the other hand, is completely different. So what I talked to you about were genetic study. You all heard about the new Alzheimer's drug, which is a monoclonal antibody directed against a misfolded protein, which is the amyloid deposition that causes the amyloid deposition. This is something similar to that. In this study, the idea is to use an antibody that will target the misfolded SOD1 protein and And in this case, this drug will be injected intravenously. This study, the phase 1 of the study has finished, and it's at its phase 2. This study only has one site in the US in California, but it's run in Canada and in Europe. The phase 2 is also placebo-controlled. So research is happening. Tofersen is the start, and there are other gene therapy coming in, and then even thinking outside the box and trying to figure out a way to stop the circulating and abnormal SOD1. Cassandra had talked about the study, I'm pretty sure. So with tofersen that can help patients with ALS, can we think about actually preventing the disease from happening, or can we think when is the best time to start? And this is the basis of the ATLAS study that has 4 parts. It starts with a part A, which is a natural history run-in. Patients are followed and their neurofilament are checked. I don't get the result either as a PI. I either know they're above a threshold or below a threshold, but I don't know what the actual neurofilament level of my patient is. If the patient reaches a certain threshold for the neurofilament, they go in part B, where it's 6 months of placebo-controlled, and after that, they go to part C, where they take the medicine. If they develop symptom, even without elevated neurofilament, they go in part D and receive tofersen. So this study, the inclusion for this study, there are only 17 specific mutations in SOD1 that are supposed to cause a very fast progressing disease. I know that it's frustrating because it doesn't involve all the SOD1 mutation, but the reason for that is this study was based on the work of Dr. Benatar from the University of Miami, and on his registry study that many of you have heard about and probably participated in. And that work, based on the patient who have a fast progressing mutation, we know about the trajectory of the NF neurofilament elevation. So we know we do know that in the specific mutation, usually neurofilament will start going up about a year before developing disease. So there is something we know. The other mutation, we don't know much about them, and that is the reason why they're not included in this study. Now, true, you can, if you have one of the 17 mutation, you're automatically in the study. If you don't have one of the 17 mutation and have another mutation, we have to do 2 things. We have to prove that the mutation causes a fast progressing disease. And we have to also prove that it's fully penetrant. That means every single patient carrying the mutation had manifested disease. And it's appearing every single generation. It's not skipping generation. So it will require actually making a very long and very extensive family history to kind of pinpoint those things. So if we are able to prove that a separate mutation from the 17 is fully penetrant and is present at every generation, or is causing also a very fast progressing disease, then it will be allowed. So beyond actual interventional study, there are a few registry study or what Cassandra said, observational study that will help understand a lot of the things that we now know about pre-symptomatic ALS. I'm citing them here. So basically, this is the pre-fall study in Miami. It is sponsored by the University of Miami. The principal investigator is Dr. Benatar, and it does require that patients do travel to the site every 12 to 24 months, and does require that you do a spinal tap, an EMG, an extensive exam, blood test, and so forth. Patients have to have a first-degree family member that has disease, and they may choose not to know about their genetic test results. This is the second registry study. It's called the DIALS. Oh no, I'm sorry. This is not the DIALS. I can see my. This is the ALS Family Project, and it is located in New York. It's sponsored by Columbia University, and Dr. Matthew Harms is the primary investigator or the principal investigator. It's very similar to the Miami one. And again, you can decide that you want to know or you don't want to know. And actually in this study, even if you are, if you are negative, not if a sibling that is negative could also participate in the study and will be a control for the patients. This is the DIAL study, the Dominant Inherited ALS Network. And this study runs in 2 sites. One is in Boston and one is in WashU. It has multiple agencies that fund it. And I don't know if this one funds travel or no. I think probably not. And again, it's the same thing, and it's recruiting. So there are 4 studies that are registry studies that are open for patients who have family members that have ALS and think they may have a familial disease, or they may be carrier of a genetic mutation. And I understand that New York, Miami, Boston, and St. Louis are not accessible to the vast majority of the country. But there is a new development, and I'm sure you've all heard about this Act for ALS, or Accelerating Access to Clinical Therapy for ALS. And it's a law that passed in December of 21. And the purpose of this law is actually to improve research and access to clinical trial and expanded access for the ALS community. And this past October, we knew that the NIH has given a big grant to develop a new national ALS clinical research consortium called Access for All in ALS. There are 2 coordinating sites, one in Boston and one in Arizona, but there will be 34 clinical sites. And they will focus on acquiring information on patients with ALS, but also on individual at risk of developing ALS and healthy control. So I'm hoping that this study will actually increase access for pre-symptomatic individual or individual who are at risk of genetic ALS to access those registry. They are very, very important. And actually, it's only through the study of the at-risk individual that we will understand how the disease starts and what are the things that may push a patient from being a carrier to actually having ALS. And I wanted to quickly add something. Patients who have a genetic mutation do not live in a vacuum. They are live in an environment, and they have they have choices that they make every single day that will impact the risk of developing disease. I wanted to kind of talk to you a little bit about this and highlight the work of Stephen Goodman, who really is interested in studying. He is at University of Michigan and at Michigan University, I'm sorry. And he's interested in the role of the environment in the risk of ALS. And I want to just show you this, for example, and this is really a parenthesis. So if we take 2 people, one that doesn't carry a risk of ALS and one that has a higher genetic burden through maybe has a genetic disease, a genetic mutation, for example, like SOD1, and you expose them to the same environment, they have the same environmental burden, you could see that the patient who have the mutation will develop disease earlier than the patient who doesn't have the mutation or doesn't have a genetic burden. If you take now 2 patients who have the same genetic burden, but expose them to a different environment, you could see that the patient who have the higher the higher environmental burden will develop disease first. In 2014, one researcher, ALS researcher, Dr. Amar El-Shalabi from the UK, used the mathematical equation that was used to determine the risk of developing cancer and used that same equation to look at the risk of developing ALS. And he established that to develop ALS, you need 6 hits or 6 steps. Usually, these are supposed to be environmental factors that will cause the disease to happen. And this study has been replicated in Europe, in multiple sites in Japan, in Australia, the same way they found the 6 steps. In Korea, South Korea, for example, you only need 5 steps. However, if you do have a mutation like SOD1, for example, the mutation accounts for already 4 steps. You need 2 steps to develop disease. If it's C9orf, it's 3 steps. If it's DDP43 mutation, you need 4 steps. So even if somebody carries an SOD1 mutation, they need extra steps to develop disease. They need other environmental factor to push them through the edge. We don't understand them all very well, but a lot of work is being done on that. So what are these environmental factors? There are many, many epidemiological studies that looked at those things. They're not great because many of them need looking back and having to answer surveys about things that were done in the past, which is very difficult to remember. And so every single one of them is not 100%, but these, for example, what we know the most. In certain occupation also, if people happen to be working in certain environment, that may actually also push them through the edge. And that's all I have for you. Thank you. And I'll stop sharing.
Hello, I believe I am next. I know that Jean's going to be sharing some pictures as I kind of talk of different pictures of my family. But I am Sarah. I also come from an SOD1 family. We carry an E100G mutation. And I was diagnosed with ALS in 2020 at the age of 26. We have lost a total of 22 family members throughout generations, and there are currently 6 of us living with ALS. So extensive history. It's taken some of the most important people in my life, including my dad and my aunt. It's always been very scary in our family, so we haven't ever really talked about it much. But recently, more and more conversations are happening. I think that's largely due to the research that's going on right now and all the excitement, and then also the majority of people that are currently living in my family with ALS. We have a lot. So it seems like, every family gathering, it's being talked about and we can't really escape it. So it would take me forever to try to go through everybody in my family who's been impacted. So I'm going to kind of keep it close to my direct kind of close-knit family that I've always grown up around. So for me, it hit close to home when my dad was diagnosed in 2015. He was diagnosed just before my senior year of college and lived for 3 years. He passed away in the spring of 2018, just right after Father's Day. So he was a farmer, hardworking man, very, very strong. So to see him kind of go through the stages was very hard. To watch him go from being a very independent person who was always helping others to being the one who needed help was very hard and eye-opening. And he was the first one in my family that we really had to kind of support through that. And he passed in 2018, and my aunt was diagnosed shortly after in 2019. And then since then, it's really kind of been one right after another. And then I was diagnosed in 2020. So from 2015 to 2020, there were at least 3 of us. And like I said, by now there's 6 of us. I was personally diagnosed in August of 2020. Prior to that, I was a runner. I worked out all the time. I had just finished my first marathon. And that's when I noticed that something was not right. I was having some calf pain, and then I noticed, actually my supervisor at the time was like, "Why are you limping?" And I didn't even know I was limping. And so in March of 2020, I went to my primary care doctor, right at the start of COVID. And then eventually went to a sports medicine doctor. I had every x-ray and MRI done. I think every part of my body has had an MRI, and we didn't have any answers. So it came to the point where I met with the sports medicine doctor, and she said, "I don't know what to tell you. You need to go see a neurologist due to your family history." So like I said, at that time, I was 26. So ALS was on the last thing on everybody's mind. Everyone thought it was a running injury. There was no way it could be ALS. And it was presenting kind of weird, the fact that, I just finished a marathon. Why wouldn't it be a running injury? So I went to my neurologist, did all the things, and then did my genetic testing. So my genetic testing came back in August. So started seeing a doctor in March. By August of 2020, I got my official diagnosis that yes, what I had going on was in fact ALS. So as you can imagine, at the age of 26, I thought that my life was going to be over. I thought I had that, what everyone says, typical 2 to 5 years. My dreams of traveling and getting married, being a mom, and running more races, I thought it was all over. I was sad and depressed and felt sorry for myself most of the time. I remember that year, especially those months, there were a lot of tears, not only from me, but from a lot of other family members. And my doctor kept saying to me, though, that ALS today is not the same as it was. But all I knew were, the people that I had previously seen, and I didn't believe her, and I didn't know what she was talking about. And then I started to kind of do some of my own research. I got connected to some organizations and connected to some people, and I learned more about the drug studies that were going on. And after reading about all the studies, I realized that this was my chance to really take charge of the disease and take control. So I decided to participate in a research trial as a way to give my life purpose and hopefully change the course of the disease for, the family that I had lost, but also the family that would be very likely facing a diagnosis in the future. I wanted to know that, I was doing something to help. So in my research, I specifically learned about tofersen and read many promising articles, knew about all the excitement around it. So of course, my goal was to get into a tofersen trial. But if I didn't, I had the mindset that I would get into any trial. I would just try them all, but tofersen was the first one. So I reached out to every Midwest tofersen site. I know there was one in WashU. I think Mayo had one. I want to say there was one in Detroit at the time. So then I got accepted, luckily, into the trial at WashU in St. Louis. And that was just 2 weeks after my official diagnosis. So I actually started looking at trials before I had my genetic results, so that by the time my results came in, I could be enrolled. So I was enrolled in WashU, and I had my first 3 visits in St. Louis. All my transportation and travel was covered by the trial, luckily, including hotel stays. So for my first one, my mom came, and we had a little weekend together. And then for the second one, I actually went alone and I loved it. I had a nice little 3-day weekend in St. Louis. And then the third one, I had another family member come with. So it definitely did help make the time kind of go. And it was kind of exciting to get away in St. Louis and explore. And then luckily, I live in Chicago, and Northwestern opened a site, and it was an easy, smooth transition from WashU to Northwestern, and I have been at Northwestern since. So bringing that together, participating in research has definitely given me more purpose. I no longer felt as sad and scared. When I first started going to the trials, I didn't really know what tofersen was going to mean for the familial ALS family as a whole. So there was some excitement, but still it was pretty new. So I really was skeptical and afraid, and I still, was afraid to make plans and talk about the future. But it did give me some peace and some hope to know that I was at least contributing and at least doing something. My mindset was if the trial didn't help me, it would at least be helping those in the future. I'm a firm believer that we learn from the successful trials and the unsuccessful trials, that they also tell us something. So no matter what, I was contributing to the research going on. I definitely adapted more of a fighter mindset, and that helped keep some of the bad thoughts away. And then, as many of you know, the tofersen trial included that open label portion. So I'm still contributing to the research. I go every 4 weeks and I meet with the research team. Some appointments include the breathing and the muscle tests. Every appointment's a blood draw, and some questionnaires. So those appointments definitely help keep me going while they are a little longer. The research team is wonderful. So they help make it more enjoyment. And it's my kind of reminder that I have to keep going and keep fighting. And I definitely will say that I know that I'm lucky that I was able to get into a trial that was this successful, and that had the open label component. So almost 4 years later of participating in the research, I've not had any changes in my progression, and I definitely have more hope now than I did 4 years ago. I'm planning for the future. I'm still traveling. I'm going to Austin this weekend. So I'm definitely, just it's really changed everything for me. And I know that this is just the beginning and there's so much more to come. It's definitely helped me to feel less in control, or more in control and less defeated. And I have so much more hope for the research world and for those participating in drugs. I hope that everyone participating in drug studies can have the same experience that I did. That would be the goal. And I would encourage everybody who's either newly diagnosed or anywhere in their disease progression or pre-symptomatic to definitely look into research, because it has changed the way I look at ALS and the way I live my life. So thank you.
Thank you so much, Sarah. If the rest of our panelists want to jump back onto video and we can see if there's any questions from the audience. There's a few that are in there already, but we'll give it a moment to see if any new ones come in.
I did see a question about neurofilament that I'm happy to answer. The neurofilament studied were the neurofilament light chain, not heavy chain. That was the question.
Very good. And if any of our panelists have any observations that accrued while watching the webinar, feel free to share those as we see if there's any more of the the watchers have any more questions.
I will say that from a patient point of view, I do feel some of the atmosphere is changing among neurologists. I know I'm some of the lucky few, but by first going through the genetic counselor, my appointment was followed up with a neurology appointment, and they took baseline lung and they took baseline EMGs and everything else, so that even without being able to access some of the other studies, if I felt like there was something going on, I could go back in and they could see if there was a change. And I think that was a really big improvement over what I've heard has been going on for the last 5, 10, 15, who knows how many years. So I encourage anyone who is, feeling scared, that if you can get the right person and the right support, you can find a way to, kind of put your mind at ease, or at least be aware of what's going on. That's all.
Yes, the neurofilament have been a game changer, honestly. I've been doing this for 20 years, and we would never even talk about pre-symptomatic. We don't even want people to get tested. But of course, with tofersen now, of course, you want to test everybody. Of course, you want to test and identify carriers so you can follow them very closely. And even though we don't have clear guidelines on how often do we want to see this patient, how often should we be doing neurofilament, how many EMG should we do? I mean, I think putting the foot in the door and being involved with a multidisciplinary clinic and being examined by a neurologist as a baseline is very important, and being followed very closely from there.
There's a question about, and Doctor, that was a beautiful comment, and we are so appreciative of it and your colleagues and all the great stuff that's happening, and that Julie's had a good experience. That's so wonderful. So Sarah, can you explain what open label means?
Yes, I can. So open label is a part of a trial so if you participate in the trial, you're automatically enrolled into open label, meaning during the trial, you were either getting drug or placebo. I still don't know what I was getting, so you kept quiet until they opened the study. And then after you complete your trial portion, so like my trial portion was 6 weeks. 6 months, sorry. So after the 6 months, you're rolled into open label, meaning you're rolled automatically for sure getting the drug, and that's 100% covered by the drug company. And like, for instance, for the tofersen trial, they gave a specific amount of time that they would do the open label. I think it's kind of changed throughout the trial, but usually there's like a specific amount of time that the open label is going on. And then I know like for tofersen, they're still collecting that data for during the open label. So did I forget anything?
Doctor, I'll defer to you if there's anything else to add. There is a question which I think is interesting, and I'm not sure if this is what they meant, but they did say, you know, I suppose, Sarah, hearing about all your travel and all the things that are happening over the years, that, you know, why would someone with a slow progression be in a trial like that? Was that not a condition?
I can answer that.
Yeah, okay, very good.
I can answer that. So remember when I showed you that the study did not only select fast progressor, they enriched for fast progression. So they had 108, 60 were fast progressor, but 48 were normal progressor. And you only needed to show a certain progression on the ALSFRS and a certain number for the breathing test to qualify. So it's the Atlas study that is really focusing on fast progressing mutation, but the Valor study did not do that. It enriched for it, but it wasn't exclusive. I hope I answered.
Another possible thing too, Sarah, stop me if you think about this too, is possibly the treatment has been, the trial drug was doing something for you that has allowed you to.
Yeah. And because if you look at just my family, the progression is pretty typical within the 2 to 5 years, with some outliers. And I mean, I guess I don't know what fast is, right? In the ALS world, what is fast? Because it all is pretty fast.
Everything is fast. Everything.
Yeah, it's all fast.
It's all fast, absolutely.
And I went, you know, from March to August. March, I went for, I attempted a run. I was able to still run a mile in March, and come by August, I was wearing a brace and had difficulty with stairs. So like, you know, everybody's different in that sense. And Dr. Driss, you can correct me if I'm wrong, but isn't the research also shows that the sooner you get it in your disease progression, the better it works. And I'm young, so there's that factor too. So I think there's a lot of factors that we still don't know fully about, that they're still learning about, that are contributing to all of it.
Absolutely, yes, you're correct.
Well, there's not any more open questions. Oh, Cassandra, please jump in. And if you could make this your final, and then if anyone else has final thoughts.
Yeah, I just wanted to, reference back to something Dr. Driss said earlier, is it used to be that families were told not to test and not to think about it. And for everyone listening today, that is not what our neurologist friends are saying. They're saying get tested, get your families involved with research. You can see from Sarah's talk that there is hope for all of us. We're all working really hard to get things changed. And I think we do have a much better future than the generations in our families that have been lost. Our group is working with a group of neurologists on a standard of care for pre-symptomatic genetic carriers. So keep an eye out for the white paper that's coming. But I just think this is a great time, and I'm so thankful for the families that were able to join us tonight.
Sarah, any final thoughts before we close? And we'll just go around the horn.
I just wanted. Oh, sorry. She might have said Sarah.
Go ahead.
I just wanted to add, I think with the Atlas trial for pre-symptomatic people, some people might really be eager to get tested because they assume that they might be accepted into the study. And that's where talking with the genetic counselors is super important to kind of learn a little bit more about your specific variant and how penetrant it is, whether it is considered one of those fast progressing variants, because you might find out that you're not eligible, and that might affect, whether or not you truly do want to know your genetic status.
Sarah, go ahead.
Oh, I was just going to echo that, especially in my family, there definitely are more and more people who are also getting tested because it is different than what it was before. And I think I have one family member in mind that, their reason for doing it was so that they could get ahead of it and, start to kind of see, be monitored by a neurologist. And if they notice any symptoms, get in right away. So I think it definitely, I kind of wanted to echo what was said earlier that, it's definitely different than it was before. Definitely different than even when my dad was diagnosed 9 years ago. So.
Can I add one thing? Although genetic testing is available and is free, and your primary care physician could order it for you, please don't do it that way. It's very important to get genetic counseling because knowing the result may not be the answer for every single person. So I'm just a plug-in for, yes, do not do any genetic testing without proper genetic counseling.
I can give my little wrap up, but I also wanted to add, in my phase in between finding out that there was a significant family history and finding out my genetic status, it did include genetic counseling for me. And like they've said, she had actually seen people that were very optimistic about what they would do and how they would feel, and then spiraled after getting. I was in a unique situation where, speaking to family members, nobody actually knew the gene. And as people know, a lot of the familial ALS genes are still unknown. So I basically went into testing blind, not that a positive would be a positive, but a negative may not actually be a negative, because nobody had known the gene at that point. But to me, it was important to me that if I came out positive, I could help other people in the family. This other distant cousin I was talking about, who was going through the same thing I was, my own children, that from now on, everyone in my family knows which gene they can look for, and that it is identified. So there are a million reasons to do it. There are a million reasons not to do it. And to me, personally, it empowered me. I was able to find a community online and find the community that started even before End the Legacy started, and then join End the Legacy and feel like I had, a semblance of control over the situation and how I could affect how we moved forward. But not everyone feels that way. So, and there's also other things to take in consideration. There's whole big landmark disputes over, genetic discrimination. There's things that genetic counselors can really help you through. So I wanted to give that little burb, and that was all I wanted to add in. And I hope everyone here learned something or had a takeaway.
Can I just add one more thing? I'm sorry. I promise it's going to be the last. If you get genetic testing and you are negative, do not think that people who are negative are going to be just happy. And there is something called survival guilt to be dealt with as well. But I want to tell you, if you want to participate in research, you could, because healthy siblings help us with that question of environment. Here we can know the environmental risk of ALS when we study sibling, for example, or other negative people that are not carrier from the same family, yet they were exposed to the same environment. So that is very powerful as well.
Well, thanks again to everyone for coming in. Thanks everyone for participating. Thank you to all our panelists. We'd love to be in touch with you, so feel free to visit our website, endthelegacy.org. You can email us, info@endthelegacy.org. And I just dropped in the chat a very quick post-event survey. We would be so grateful if you filled it out. And we'll also send that in an email. So with that, we will close for tonight. If you know folks impacted by other genes, we're doing C9orf72 next Tuesday, progranulin the Tuesday after that, and we're working on scheduling a MAPT1, but ultimately we'd love to do one of these for every gene so that everyone can have an accessible information like this from the comfort of their home. All right, everyone. Thank you so much again, and we will close for the night. Thank you.
Bye.